And now it's time for our next speaker, who is Viveka Nordberg, a neonatologist at Karolinska Sjukhuset, chair of the NGO Paediatric Health Initiative, and a PhD in neonatal infectious diseases with focus on antibiotic resistance.
Yes.
Is that correct?
That is correct. Thank
Great.
you so much for the presentation.
Welcome.
Thank you. So, just a quick look on this one.
This is to move forward.
Yeah?
This is the point, but they don't recommend.
Okay. So, thank you for inviting me today. And I'm going to talk about some new strategies to identify virulent and resistant bacteria in neonates in low-income countries. It's a part of a bigger project working with evaluation of the possibility to prevent neonatal sepsis, as you understand. So WHO urges countries to adapt to responsible rational use of antibiotics. And how do we do that? Well, first you need to tackle the threat of antibiotic resistance in both very intensive ways and as well humble ways, to see in what setting are you and what can you do about it, and also to strive to reach the SDGs. So in order to have this approach, what do we need? Well, we need really fast, easy methods to get detailed information about bacteria. It's just not talking about E. coli Klebsiella. What kind of Klebsiella is this? What kind of attribute does this bacteria have? So what is there then? It's also very nice to talk about what's already there. And since some years back, there are teams around the world working with this. And the team that I've been building and working together with are from different parts of the world. Here we see some of our colleagues working in the laboratory in Uganda, in the laboratories in Tanzania, in laboratories in Bath in UK, and as well here in Stockholm and in Gothenburg. And as here we also see Peter Weiswa, because he's involved from Uganda. So these are key persons working with microbiology, biophysics, biotechnology, and neonatology, because as Joyce said, you need to not only love the biotechnicians, you need to love the biophysicians and all the people that knows a lot about their things. And we've been talking about developing methods that are really complicated to make them work in an easy and fast way. And when working with multidrug resistant bacteria as outbreaks, as you talked about, Joy, we know that the dissemination of multidrug resistant strains, that is really a risk for subsequent invasive sepsis. And you don't want to, you need to know what kind of bacteria that is there. You cannot just treat with antibiotics and think that you're doing the right thing, because that's what's the base in antibiotic stewardship, right? So local surveillance is the key. If you're working in Sweden, you need to know what's around here. If you work in Ginger, or if you work in wherever place, you need to know what bugs are around, and this in my ward. So in order to control outbreaks, you need to do good diagnostics, because they can really curb the spread. So that is the base of treating the right bacteria, or treating the right neonate for the right bug. So that is what we want as a base in antibiotic stewardship. Also going for the good microbiota in the developing neonate. So needed interventions then to decrease sepsis-related mortality. We know that the research gap is that most of the diagnostic procedures to find the bacteria are very complicated. They need, first of all, they are culture-based, and they need quite some advanced molecular sequencing methods to know what resistant gene is this, what kind of virulence factors does this E. coli have. And they take time in Sweden as well. It takes 2 weeks to do all this, sequence a Klebsiella, for example. It costs a lot of money. So we need to round that. We need to skip those steps. And if we do that, we can find virulent bacteria and also the resistant ones. But we need to know, where are the resistant genes in the bacteria? Well, the resistant genes in, for example, the gram-negative intestinal bacteria, they are in the plasmids, and they can sound very complicated to find the plasmids, but if you want to know about resistance, you need to go to the plasmids, because the genes are not in the chromosomal DNA of the bacteria. So you need to find an easy way to look at the plasmids. And extracting plasmids could actually be quite easy if you know the steps to go, and it does not take a very long time. So if you look for the plasmids in the resistant bacteria, you can also manage MDR in a more easy way. So how do we do this? Well, one part of the projects that I'm working with have been, since some years now, been developing 3 different methods. The first 2 is about looking for MDR in gram-negatives. And the third one is to diagnose GBS really fast. So they're designed to work in low-resource settings, and these protocols will work in existing infrastructure. No advanced machines. We're talking about having an oven and having a quite easy microscope, as we see here. These microscopes, you diagnose malaria, for example, in most hospitals around the world. So also putting this into a low-cost technique, I think that's a good way to walk. So since I have very, very short time, I cannot tell you exactly how these methods work, but for very short, I will tell you that the first one is what we call PNA fish, and that's PNA is a synthetic peptide nucleic acid that complementary binds to specific regions in the bacteria you want to look at. So you make the probes for these, and those that we have been developing are for some specific E. coli and Klebsiella strains. And they're also marked with fluorescent labels, and we can then identify virulent bacteria, the E. coli and the Klebsiellas. We know that these bacteria that you see here, they stand for at least 80% of all neonatal sepsis in the world. So those are the guys we want to see. So you will have the answer in 90 minutes if this baby is colonized with a virulent resistant bacteria. And it is a low-cost technique. You don't need to do any cultures, and you can do it in a low-resource setting. So the next one, cut and stretch assay. You remember the resistant genes? They are on the plasmids. So we will cut the plasmid and we will stretch the plasmid. That's what this method is. And when we do that, we will find the resistant gene. And using the restriction enzyme, CRISPR-Cas9, that got the Nobel Prize 3 years ago, we can label that CRISPR-Cas9 for the specific gene. You know, the cefotaxamases, they have CTXM, the carbapentamases have the NDM1, for example. And we put them into the same test tube as the plasmids are, and if there is a cut, the plasmid will become linear. And then we will dye it and look at it in the microscope. And we also developed a microscope that you can just attach to a smartphone, of course. It's the other smart guys, it's not me. So doing this, we will get a rapid genetic information, skipping all the molecular sequence analysis, and we will get the answer within 2 hours. So you can have this as your outbreak detection. So the last method then is a group that we work with in Bath in the UK. It's another method that will give us the answer about if the mother is GBS positive or not. And using a biosensor with vesicles that are filled with fluorescent markers, we can find in vaginal smear in the mother that comes to give birth at the moment. We cannot do it before because we don't know where she is. But when she's there, we can do this. And when the GBS is touching this vesicle, it will lyse and all these fluorescence will come out. And within quite not so many minutes, 45 minutes, they say, we can see if this mother is GBS positive or not, and we can treat her with that antibiotics that she needs, not this broad spectra that you give just in case. And of course that, as you understand, is a point-of-care test, bedside test, takes 45 minutes and will be a base in antibiotic stewardship. So these tests, they are tools for what we want. We want the outbreak detection. We want to see how big is the burden of antibiotic resistance in this ward. We made some point surveillance studies already, both in Tanzania and Uganda. We see that the prevalence is very, very high. It's around 80% of the babies colonized with ESBLs, for example. We can use this for sepsis prevention. What mother has GBS? And we will also look at E. coli and Klebsiella in urine for birth-giving mothers with using the PNA fish. This is my last slide. And what are the challenges we need to overcome in order to make this new technology available in low-income resource settings? Well, we first need to show that research, and we need to show that the evidence that demonstrate that this technology actually it has a potential. And doing that is by collaborating with academic institutions from there and here, from around the world, and in parallel with educating the staff, the laboratory personnel, we will take the steps forward. For example, this Saturday, a microbiologist is coming from Tanzania to be in Sweden to do all the PNA fish and all the catch and strays for 3 weeks. And that's one of the parts of the collaboration to get forward. And then the policy changes that promote the use of this test will also be very important. Thank you.
For your speak, Viveka. I think we're running a little bit late in our schedule, so I think if you have questions, we can save them for the break, both for Viveka and for Mats. And we welcome our next two speakers.